happening. And then you get a bunch of activation because of that. And then also what's really interesting is that because of all of this blood loss and because of the course of care, these patients are then systemically hypoperfused, right? So there's lower volume of blood, so you get, there's just less systemic perfusion, and therefore the endothelium becomes activated, you get release of cytokines and DAMPs, and all sorts of a milieu of a mess of things that make these patients incredibly complex. And then again, as I said, this is modulated by the blood products they're getting and also can change while they're in hospital recovering. So they can show up as unable to clot and then have too many clots all at the same time. And what I'm really interested in is understanding how we can treat these bleeding patients that have coagulopathy. So we give them blood, right? We give them blood products, we give them transfusions, but those products, we can actually think of them as having their own coagulopathy, right? They're blood that's stored in a bag. And some products, I'll talk a lot about platelets. Platelets are stored at room temperature in their current licensure for 7 days. And you know, if you work in the lab, if you can imagine taking something biologic and putting it at room temperature on the bench for 7 days, it's going to be pretty gross at the end of 7 days, right? So they have a unique storage lesion that manifests over the course of the time that they're in the bag, right? And so you can see here some changes in stored red cells. You get a kinocyte formation. And red cells are stored for many, many weeks, right? They're stored for up to 9 weeks, if not longer. And then platelets, you get platelet activation during storage. You get all sorts of receptor interactions. Platelets are also stored in plasma, so you get platelet-plasma interactions. And over time, there's biochemical and functional changes in these products that mitigate their efficacy. And we don't fully understand the storage lesion, and we also don't fully understand what the storage lesion does when you give this product to a patient. So what is the role for bioengineers and you know, folks like me and those in this room to contribute to this? And so, as you know from the title of my talk, we use microfluidic models. Why are microfluidic models important? Well, hemostasis, this whole process that we're talking about in these injured patients, is very flow dependent and has mechanisms that are very specifically flow dependent. So hemostasis is the stopping of blood flow, right, manifesting in a clot, which we all understand and can think of manifest a clot. But in the body, where you have these different settings of the vasculature, depending on the flow rate, you get challenges between convection and concentration. So say you have an injured vessel, the process of coagulation is trying to generate enough protein and enzyme to achieve a clot. And that's a cascade, that's a canonical correlation cascade that requires these high concentrations of protein. If you have high flow, those proteins are going to get convected away and you're not going to be able to achieve clot. And so we actually have compensatory mechanisms to initiate hemostasis at these high flows. And von Willebrand factor, this protein here, is central to that. So it's a shear activated protein. It circulates as a globule, and under high shear only is it activated to elongate and extend, which enables capture of platelets to initiate hemostasis. If you are not testing hemostasis in a flow setting that potentiates this mechanism, you will not capture it. And so this is really important to consider. And then in addition, blood is a really interesting fluid, and the biorheology of blood is flow dependent. So it's a non-Newtonian shear thinning fluid. That means at lower shears, it has higher viscosity than at higher flow rates and higher shear rates, which is because of the red blood cells. And this is really related to that concentration and deformability of red blood cells, which I'm sure, as many of you know, can be modulated by disease states, including bleeding. As we said, trauma patients get anemic, they get acidotic. So this is all potentially changing their biorheology. And all these other unique aspects must be considered when we're thinking about understanding hemostasis, testing hemostasis, and designing devices. If you don't appropriately capture and consider these mechanisms, you're going to miss something when you're trying to understand what's happening in your patient or in your blood product. And the last flow-based mechanism I'll talk about is margination. So blood, as we know, is a colloid. It's a suspension of cells in protein, salt, and water, right? And because of those red cells, they actually marginate platelets to the periphery. So just by nature of the structure of our blood, you're actually primed to initiate hemostasis because platelets, which are the first actors in hemostasis, are closest to the wall. So again, if your model or if your system doesn't recapitulate this, you might miss some flow-dependent mechanisms, and you therefore might not be appropriately capturing hemostasis and able to measure coagulopathy appropriately. Contrasting that, clinically, hemostatic testing does not encapsulate any of those mechanisms. So there's a range of tests that our clinical collaborators have available to them to diagnose bleeding patients and diagnose these disorders. And cell count, so just counting the number of platelets and counting the number of red cells and the amount of hemoglobin, is actually still to this day the most common test used to indicate transfusion, even in bleeding patients. We don't have a better assay, a better test to understand hemostatic dysregulation and indicate transfusion than counts. And I think we can do a lot better than that, but there are many challenges that are why we have gotten here. Plasma protein concentrations and plasma-based coagulation tests are some of the most common tests used to diagnose hemostatic disorders. So this is just measuring what's in the plasma and also initiating clotting in the plasma and measuring clotting times. As you can see, based on all those mechanisms I just described, it's agnostic to that. There's some platelet function tests, but these are, again, mostly stagnant. There are some not stagnant platelet function tests, but they don't work very well in bleeding patients and they're not widely used. There's viscoelastic tests, which are the devices shown here. So they cause a clot in a small, in a cup, or in this case in a cartridge, and then they measure the mechanical properties of that clot and pull out different parameters about the mechanical properties of those clot. And that's one of the, in bleeding patients in particular, is associated, dysregulation in these parameters is associated with poor outcomes. And there's research use only tests. But really there's no widespread flow-based assay that's used to diagnose bleeding disorders, and especially not in bleeding patients. And when patients are bleeding, this is a recent modeling paper, we know that that bleeding is occurring at really high flows, right? We're talking about transected vessels and we're talking about injured vessels. So the flow rates, and especially if you have an injured artery, right? The flow rates of injury and where hemostasis is occurring in these patients is incredibly high as heart attack and stroke and stenotic vessels in the body. And again, all of these clinical assays do not recapitulate this. They are all stagnant. So we have no idea if these mechanisms in these patients that are exsanguinating are dysregulated. And again, summarizing, they just tend to be stagnant, they lack biological surface, and again, a widely used assay has flow. And so these are major gaps that can be filled by microfluidics. And so we use microfluidics as a tool to provide better insight into these mechanisms and recapitulate all of these pathophysiologically relevant mechanisms to study both patients and dysregulated platelet function and coagulation, as well as the treatments that they get, including blood products and transfusion. And I know there's lots of folks here that work on microfluidics, and so I'll summarize pretty briefly. A lot of the strengths of microfluidics are that they're completely customizable. If you can draw it, you can probably make it in a microfluidic, right? Small volume requirements, you can get assays down to hundreds of microliters. You can match the flow and capture all of these flow-based mechanisms. And you can also cater this to your specific pathophysiology of interest. And then because you can do immunofluorescent microscopy, you can extract a multitude of these mechanistic parameters. So we can provide a lot of mechanistic insight. And so our group uses these tools, again, to study trauma and transfusion medicine and other bleeding patient etiologies to provide more insight into dysfunction and dysregulation, and ideally make better treatments for bleeding patients. So I'm going to talk about a few ongoing studies and stories from our group that we've done to either characterize blood products or model transfusion at the bench. So first I'm going to talk about platelets. So as I mentioned, platelet products are licensed to be stored at room temperature for 5 to 7 days. In the United States, we collect platelets via apheresis, and I'll talk a bit more about apheresis. In Canada, they're mostly whole blood derived platelets, so they're made from centrifuging platelets out of whole blood units and pooling them together, majority products. But again, any stored product acquires a storage lesion. So as these cells and plasma and electrolytes are sitting in this bag, they are, they're generating lactate, they're metabolizing, they're trying to keep themselves alive. And as they're doing that, they're changing the biochemistry of the bag, and they're also consuming their own functional abilities and consuming plasma in the bag. And because room temperature platelets have such a short shelf life. Many centers can't even afford to have platelets. They're too expensive. And while we also have a blood product shortage because we don't have enough donors, we also waste over $80 million in platelets every year in the United States that gets thrown out because they expire. And so it's a really costly inventory to maintain. So what we've done, and our group has looked at this, and other groups have too, is you can store platelets in the cold and potentially increase their shelf life. And what's really interesting about platelets is it doesn't just slow down the storage lesion. Platelets actually have receptors that are sensitive to temperature, and so their storage lesion completely changes when they get put in the cold. So they not only last longer, but they have different mechanistic properties that make them actually more pro-hemostatic. They have less circulation time, which is why when this was originally investigated in the 70s, people just said we need longer circulating time, so we're not going to have cold platelets. And now there's been a big resurgence in use of cold platelets for bleeding patients, and because it makes such logistical sense. So in the first study I'm going to talk about, we just measured the hemostatic function of platelet products using microfluidic. So this is a microfluidic device. It's a pretty straightforward device. It has it's a straight channel, parallel plate chamber style, and it has a narrowed center. And what that narrowed center does is it creates a region of high shear stress that potentiates that von Willebrand factor capture of platelets and allows us to measure the hemostatic function of this product. So what you're seeing in this video is just a transmitted light, a phase image of this platelet product forming clot in this microfluidic channel. So this is the product alone. The platelets are stored in plasma. They have everything they need to do to make this clot. And then what we do is we measure the outflow. So we're perfusing this system under constant pressure, under an arterial pressure. You perfuse the product through the system. If it clots, the flow rate slows down. And you can see that here as it peaks out. And so we use this system to measure the function of these products and compare the effective storage temperature. And before I get into that data, when you use these tools, you can also do immunofluorescent microscopy, as I mentioned, and you can really see when we add a label for CD41 here, that there are these distinct populations of platelets in the bag that have shed receptors, but are still able to, they're able to bind collagen under high shear. But there's lots of different platelet populations, and some are functional and some are not, manifesting themselves over the course of storage. So again, we use this model to compare temperature storage of platelets. And we stored both products out to day 21, 3 weeks of storage, both room temperature and cold. And this black line here at the left is what the product looks like on day 0. And using this microfluidic model, you can see that cold stored platelets, even out to 3 weeks, have better flow profiles, so shifted to the left, closer to baseline than their room temperature counterparts. And when you get to 3 weeks of room temperature storage, you essentially have a straight line, meaning no clot is forming at all. It's just constant flow. So we used this model to, one, just confirm that these platelets at 3 weeks in the cold have the ability to bind under flow, and they do. And then comparing to the current licensed product. And we measure the area under these occlusion curves, and you can see they're conserved against baseline. And then we also measure the rate at which the channel occludes. And you can see, again, cold better preserves this function, especially out to 3 weeks. And using our image analysis tools and taking these images over time, we can also look at clot surface area. So we just thresholded these images and generated clot surface area over time. And then you can get things like lag time and growth rate, metrics that we're all pretty familiar with in clotting assays. And you can see, again, cold stored platelets have a shorter lag time, so they're clotting faster, supporting this better occlusion time. But the growth rate here is actually similar. So they're clotting faster and they're depositing a little quicker, manifesting in that occlusion time. So what we did with these products is we looked at the metabolomics of those stored units, stored at room temperature or cold. And I think not surprisingly, you completely change the metabolome when you store platelets in the cold, which is manifesting in this better function. And so what we did is, and you can see here these different groups, the different storages, room temperature on the left, cold in the middle, and then room temperature for 3 weeks on the right, a completely different product than these other products. And we looked at the different pathways that were most significant during cold storage, and we actually related these back to function. So we used our microfluidic model to correlate the changes in metabolome with what pathways were most relevant to flow-dependent function, and identified that taurine metabolism was really a critical part of relating to function with changes in metabolome over storage. And so what we did with that omic result is that we added taurine to the bag during storage. And if you play with that metabolic pathway, you can actually better preserve platelet function over time. And so we see here in the same assay, you get the shift to the left when you store these products in the presence of taurine. So on top of that, we're just trying to understand what's happening in the bag. In the United States, there are multiple ways by which you can manufacture an apheresis platelet. And the two main machines are shown here. The left is one vendor and at the right is the other. And despite the fact that you can see immediately the flow and the residence times and the materials are completely different between these machines. And if you know that platelets, as I described, have these shear activated, and blood has all sorts of shear dependent and material surface dependent mechanisms. These products are very different when they come off of the machine, but clinically they're treated ubiquitously. So the clinicians ordering platelets often don't know what product they have in their blood bank, and the difference in function is not appreciated or understood at all. So I'm the PI of a study that's partnering with a clinical trial. So the clinical trial is looking at cold stored platelets in patients, and we're not actually doing anything with the patients or patient samples, but what we're doing is manufacturing these products as they would be made for transfusion, and we're deeply characterizing all of the different ways in which you can make a platelet in the United States. And what's under the hood of these fluidics is a big centrifuge. So these devices have this huge centrifuge, and that's what's actually separating out the product. So in pheresis and we're making platelets, you pull off that plasma and platelet part once they're centrifuged, and you actually return the red cells to the patient. You can also imagine that these two vendors that have very different flow patterns and kits when they're pulling the blood out also have very different centrifugation strategies. They have different sizes of centrifuges, they have different rates of speed, and they have different separation strategies. So all of these things really manifest in very, very different products, yet no contemporary comparison of the function of these products existed until we started looking at it. So I'm gonna go through a lot of different assays. So as I said earlier, we have no real standard platelet assay. So when we were tasked with doing this work, and this work is going to be submitted to the US FDA, we essentially are doing a massive panel looking at all the different ways to measure platelet function. So first, this is aggregation ability. And if you see at the top right, if you're not familiar with light transmission aggregometry, essentially you just have platelets and cuvette. This is a stagnant assay. You add agonist and they aggregate. And you use light to measure the rate at which these platelets aggregate, getting an idea of their aggregation ability and function. And then from left to right are all of these different ways to make platelets in the United States via apheresis. So Trima is the machine that I showed on the right. Amicus was the machine on the left. And those platelets can be stored in plasma. So they're stored in the donor's plasma, autologous plasma, or they're stored in suspensions of mixed electrolytes and plasma, platelet additive solution or PAS. And just looking at these four groups, so two different machines, two different storage modalities and storage solutions, they're not the same product when they come off of the machine, and they're not the same product after 3 weeks of cold storage, right? This one group at the right, Amicus platelets stored in platelet additive solution, have reduced aggregation abilities with both agonists, and this is persistent over storage. And both groups decline, and I don't have this data for sake of time here, but this is actually enhanced aggregation compared to the current licensed product room temperature counterparts. So again, these are very different products. And then when we look at viscoelastic testing, so again, that's the assay I mentioned that makes a clot and measures clot strength, and we look at the maximum amplitude that this clot was able to achieve, which is known to be very related to platelets. Again, we see these massive differences in these products. And not surprisingly, these products with less plasma have less clot strength, right? But again, over time, the way that we are manufacturing these products manifests in very different functional profiles. And then I think one of the last functional tests I have here is thrombin generation. So this measures the ability of those platelets to generate thrombin. And again, we're just testing the products themselves. This is a product that would be transfused. And as you can see on the left, when you just look at the bulk thrombin generation, there's not a lot of differences in the total thrombin that these products are able to generate. However, when you look at the right, the kinetics are incredibly different. And we have that one group again that has less thrombin generation than their counterparts. And these are collapsed over all 3 weeks of storage. Thrombin generation, as you can see at the left, is pretty stable. But that one manufacturing method in combination with its additive solution make a completely different product that's not able to generate thrombin in the same way. It's a slower and more denuded process. And what's really interesting is that group that had reduced aggregation and reduced thrombin generation also has the highest platelet count. And so the current licensure metrics for platelet products in the United States are pH and count. And those things tell us nothing about function. I think historically, we thought we all, and I was surprised myself going into the study, we think pH is just speaking to the health of the product, right? Higher lactate, more metabolism, you would have less functional product. And also you probably think that it's better to have more platelets in the bag. But if they're not functional and you're bleeding, do you really want them? Or would you rather have less platelets that can clot better? And so these products are completely different. They have different functional profiles that, again, are not captured at all by the current regulatory processes through which these products are licensed. So I'm going to switch gears a little bit and go back to our patient and work towards how we can put two and two together. So we have these different platelet products and we have different metrics and mechanisms. And now we need to go back and figure out how we're going to treat our trauma patients. So this platelet piece is a really important part of trauma. Platelet dysfunction is it's critical. Every trauma patient shows up in clinic with denuded platelet aggregation ability, but we can't accurately measure it, and we don't really know what's going on. Prior to our group, there were 2 papers that I'm showing here that looked at trauma patient samples under flow. And one at the left is from the UK group from Paul Armstrong and Karen Brody, and the one at the right is from Scott Diamond's group at UPenn. And again, at the left, they had a really injured cohort. So you can measure how severe somebody's injury is. We give them, it's called an injury severity score. It's nothing that you can use prospectively because it's all done in post. But they had a very injured cohort, and you can see again that these trauma patients have less ability to adhere to collagen and make clot. And then at the right, the UPenn group had a much more varied trauma patient population. Some of them were more injured, some of them were not. And as you can see there in that array, some patients had really robust responses and some patients had platelet dysfunction. So a big mixed bag, and again, still very important to continue to understand. However, no microfluidic model is really designed with trauma and transfusion in mind. And so when we're thinking about hemostasis, right, one of the things that our group thought was this ability of the clot to make a seal and prevent flow from escaping an injury was the important endpoint. And so we started thinking about how we could design a system that recapitulated this sealing of a vessel. And there's a few other groups, and I'm showing them here, that have designed these systems. So at left, this is a model out of Wilbur Lamb's group that's endothelialized, and they made actually a microfluidic valve, and you can pop open the valve and injure a vessel. And they use that to look at phosphatidylserine exposure and some genetic inherited disorders. This group in the middle, this is Keith News' group out of Colorado. And so they made this ladder system where they're flowing blood this way, and this wound is this ladder rung going to the left, and you can see that platelets and fibrinogen fill this wound, and that group is really focused on, again, genetic disorders and hemophilia. And then at the bottom, this is a Scott Diamond model, and they were really interested in understanding thrombin flux. So they designed this model to measure flux while a clot was forming, and they made these posts to mimic the extravasculature. And this is our group's model at the right. And so our model is pretty simple, but I'll show you what we've been doing with it, because when you're thinking about trauma and transfusion medicine, you have maybe different design constraints, and you need to be able to transfuse your model. And so I'll talk a bit more about our model, but we have flow coming across the right at the top. Flow exits out of this injury in the vessel. We coat up to this point with collagen and tissue factor, mimicking the extravasculature. And then you get a hemostatic seal that's platelet rich that plugs this hole and prevents flow out. So one of the first things we did with this model was study, characterize healthy controls so that we could compare with trauma samples. So this is, again, another view of a healthy control sample, and the model design is at the top right. So flow goes from left to right through what's mimicking a native vessel. It escapes this injury point, and then this whole area is coated with tissue factor and collagen, again, mimicking the extravascular space. As you can see in this video, you get clot happening at this point of injury. It's platelet rich, and you get continued platelet activation, retraction, and strengthening of the clot to shore up hemostasis. And a healthy patient or healthy donors clot within about 5 minutes in this model. So when we compared those patients to trauma patients, and when we looked at flow both in a venous regime and an arterial regime, in both cases, trauma patients had severely impaired hemostatic ability under flow in our model. And we looked at both the bleeding time, and you can see there's many patients that don't clot in 20 minutes. And then we also looked at the frequency of the sample and closing the channel. And you can see, again, especially at high shear, these patients have reduced ability to clot this model. And so we used our immunofluorescent microscopy, and we looked at what was happening over time. And at the top left is surface area. You can see that in the healthy controls, these top lines, you get this robust, really quick surface area coverage that is platelet rich. And then if you are injured, you have some surface area coverage, but it's a bit slower and a bit flatter, and it's not as platelet rich. At the right, this is the platelet content. So these trauma patient clots have virtually no platelet content. They're depositing other things in order to try to achieve hemostasis, but it's much less efficient and does not seal the hole. And then again, we can look at these parameters. So the lag times trend higher in trauma. The growth rates, actually, some patients have high growth rates, some patients have low growth rates. And then again, they all have this reduced platelet function. And so these are what some of these trauma patient samples look like. So here's one example. You can see at left here, this clot morphology, even just in phase, is wildly different. It does not look like a healthy sample. And then you can see at the right, there's some initial platelet deposition, but those platelets the clot is not retracting, and the platelet signal completely wanes away. So these platelets are shedding their receptor after they're in the clot, unable to participate in canonical hemostasis. And then we also had some patient samples, as you saw in the data, that didn't clot at all. So they're in the same model, and there is virtually no function. So we took our microfluidic parameters, we took clinical parameters, and we did an exploratory hypothesis generating correlation study and focused really on what was correlating with these, with our microfluidic parameters. And we saw the venous microfluidic hemostasis performance was dominated by what the protein content was. So it was most high related to how much fibrinogen and other proteins were in the blood. And then really interestingly, we saw this arterial microfluidic hemostasis correlated with oxygenation status, but not in the way that we would expect. It was actually inversely related. And it turns out when you have low oxygen content, you actually get platelet activation as probably a rescue mechanism. And then again, plasma-based tests did not correlate with these same parameters. So when you use these models in conjunction with clinical assays, you're able to pick up these different mechanistic signatures that you would not get with the current clinical testing profile. And in our model, we also looked at how severely injured the patients were. And hemostatic function was not related to how severely injured the patients were, which is surprising and not what we expected. But there's actually some pretty good precedent with this. As I showed you earlier in those two studies, it turns out that really once you get into a very severely injured patient cohort, you get uniform loss of function. But if you're below a very severely injured cohort, you have a lot more variability. And there's another group that looked at the platelet transcriptome in trauma patients. And again, it wasn't necessarily injury severity that dominated their trends, but it was the type of injury that they had sustained. So after this study, we are continuing to look at this, and we have an ongoing longitudinal study. So we're taking samples from trauma patients at arrival, and then we're following up with them for 5 days in hospital. And as I mentioned, trauma patients do a lot of interesting things when they stay in hospital. And these are just some preliminary results. And as you can see, trauma patients actually show up pretty normal with pretty normal platelet counts. They're not usually thrombocytopenic. And this is dropping over time at our institution. So over the first day, if they stay in hospital, they get really low platelet counts, while at the same time, they show up with reduced platelet function. While their count is dropping, their platelet deposition is actually going up in our model. And this is correlates with the clinical observation that these patients this is when they are at most risk for thromboembolic complications. So this is the time in which they are getting DVT and venous thrombolism in hospital. And so what we think is happening, one, post-injury, you have all of those damps and cytokines. You also have ultra-long von Willebrand factor circulating, and you also are making new platelets. And so there's a new cohort of platelets being made, and younger platelets are more hypercoagulable. They're more procoagulant. So we're thinking that's what's happening here, and we're continuing to investigate that. And then the last bit of my talk, I'm going to talk about our models ex vivo to simulate resuscitation. So we use the same model, and we can either take a patient sample or we can induce a coagulopathy, and then we can simulate transfusion at the bench and measure the ability of the transfusion to improve function. And so broken down a little bit more, and typically what we do is we take blood from healthy volunteers, myself included, and many of the students in our lab donate very frequently. And then we induce a coagulopathy relevant to trauma. So we can make the sample dilute. That's a very simple representation of a dilutional coagulopathy, that iatrogenic coagulopathy I mentioned. We can inhibit platelets, so we inhibit P2Y12 with ticagrelor. We can also make samples lytic and capture these diverse phenotypes relevant to trauma-induced coagulopathy. It is a contrived, simplified version, but we are able to do this in a high throughput fashion because we're getting samples from healthy donors. And then what we do is we add products. So we've done this with, I'll talk about a couple different studies, but we've done this with plasma products, and we're doing this with whole blood, and we're doing this with platelets. And what we do when we test these samples in our microfluidic device is we take the sample from the donor, or the sample from the recipient, the healthy donor on this day of experiment that becomes our recipient in the model, and we label their platelets or we label their plasma with one fluorophore, an antibody conjugated to one fluorophore color. So in this case, cyan. And then when we take the product, we use a different fluorophore with the same antibody. So that way we can label populations from both the donor product and our simulated recipient. And then we run these in our model, and we run other tests on them. So as I said, one product we were interested in was if you gave concentrated plasma compared to fibrinogen. So in trauma, these patients get hypofibrinogenic, so they lose fibrinogen. You can resuscitate fibrinogen with plasma or with concentrated plasma, or you can give fibrinogen concentrates alone. And we hypothesize that if you give concentrated plasma, you're giving von Willebrand factor, you're giving factor 13, you're giving all of these other coagulation factors that are really important for hemostasis versus fibrinogen alone. So we did this in our model. We induced a dilutional coagulopathy, and we gave cryoprecipitate, or we just gave fibrinogen. As you can see, as you might expect, that VWF content shown here captures platelets faster and has better hemostasis than if you just give fibrinogen, right? So that's confirmatory of this idea that you're consuming more proteins than just fibrinogen if you're low on fibrinogen. And in this model too, we made a very lytic model, and that actually was so severe that it didn't clot at all in our microfluidic model. You can see it here at the left. You get some initial deposition, and it literally just gets lysed away. And in trauma patients, if you have lysis of this level, which we validated using a viscoelastic test, these patients actually have a 50% survival rate compared to patients with normal lytic activity. And so we're seeing that in this model that this much hyperfibrinolysis is so oppressive that in our microfluidic model, these patients can't even make a clot. And so the last study I'll talk about is we've been simulating platelet transfusion in this model. So again, we have a recipient that we make dilute. This is a dilutional model in this case. And then we add donor platelets, and we label the recipient platelets with one color and the donor platelets with another color. And that way we can look at and quantify both contributions of donor platelets and recipient platelets. And we can do this now to compare these different manufacturing methods of platelet products. And what we see is this really integrated clot, right? These donor platelets and recipient platelets are actually working quite well together. And donor platelets, when these dilute samples, actually recruit more recipient platelets to the clot and work really nicely together to achieve hemostasis in our device. And so what we did is we took these two populations, and you can measure the intensity of these populations, but we also just measured the surface area of each respective donor versus recipient platelet population. And what we found when we look at platelet storage over time is that if you look at the top, these are room temperature platelets in this model. At day 0, you get some really nice donor platelet deposition, and you get some really nice recipient platelet deposition in surface area. But if you store that platelet for a week at room temperature, you get virtually no donor platelet deposition 1 week later, and you get a decrease in the recipient platelet deposition in this model. On the other side, if you look at the bottom, these are cold stored platelets. And if you look at day 0 and a week later, those actually map really closely together. So cold stored platelets stored for a week are still participating in hemostasis in our model, and the recipient platelets are also still being recruited to the clot. If you store them for 21 days, again, you get this little decrease in donor platelet deposition, and you also get a concomitant decrease in the recipient platelets given. And this, again, is a dilutional. We're simulating transfusion here in the microfluidic model, and we're using this to compare these different ways to manufacture and store platelets. And what was really interesting and surprising to us is that if you store platelets and electrolytes, which typically, as you saw in all of our other data, have reduced hemostatic capacity if you measure the product alone, when you transfuse those platelets in our model, they actually do better. And platelets stored in electrolytes have increased responses over storage and recruit more recipient platelets to microfluidic hemostasis. And the same was true in the cold and persisted for up to 3 weeks. And we don't know what's going on here. They have less plasma. They're receiving less plasma in this model. They have a different biochemical and electrolyte composition, but either the extracellular vesicle content in the bag or this ability of these platelets to preserve their flow-dependent functions, because again, you don't pick this up in a flow lacking model, have changed the storage capacity of these platelets and their hemostatic efficacy in our simulated transfusion model. So in closing, I'm going to return to, I think, the big gap, a big gap in the field, and talk about clinically available flow-based assays and these challenges that remain. So as I said, there are clinically available flow-based assays. And why have they not yet solved the problem, right? Because we're making devices, and I'm an engineer, so by nature of my existence, I'm making devices, right? And I'm trying to build tools to solve these problems. So why shouldn't we commercialize these and take them to clinic? And I think the way that these devices have been developed to date have been based on dose responses for known drugs, right? Like the PFA 100 was designed to titrate aspirin doses and so on and so forth. And when you take them to a bleeding patient, that all gets lost, and you don't know what a lack of function means. And we also have a need to oversimplify the readout, right? These devices, once you take all of that data I just showed you, you have to make that into one number or two numbers maybe, right? That your clinical counterpart could use to make an informed decision about their patient. And then I think a big fundamental issue, as I've been talking about trauma, and hopefully I've convinced you, is that there's a big lack of understanding of the bleeding patient disease state. And so then we don't know how to interpret these assays when you have a readout. So again, there's still these major gaps that remain. And I think microfluidic hemostasis will be transformative, but we have a lot of building blocks that we still need to fill in, both with respect to the patient and also with respect to what you do with your device output. And our strategy, as you have seen, is to go forward. So we're trying to validate things with patient outcomes, which is one approach, versus doing dose responses or going backward to animal models. And so I think especially in the hemostasis space, we're talking about big implications, right? This is not just saving lives and caring for patients. This is also a regulatory question. And then that becomes a product design question, because you're going to design a product to fit a regulation. And so there's still, like I said, these huge gaps that we can work on in trauma and transfusion medicine. And I think this a translational multidisciplinary approach is the only way that we're going to get there to make differences and have better tools with which we can treat our patients better and design better products. And so that's really where my lab lives. It's at the center of all of these different things. And we have, as Jay said, we have lots of other projects going on in bleeding patients and trying to understand the intersection of all of these interesting spaces. But I really think a strength of these tools, of these microfluidic models, is that we can apply them in all of these different directions. You can use the same tool to better understand your patient and also to better understand your product and the patient and the product together. And that's it. And so thank you so much for coming and listening. And I have a lot of really great collaborators and a really great team back on the other side of the continent. We're working really hard to solve some of these translational problems. So excited to be here and meet with you guys today and see what you're doing too.
Thank you very much. Questions for maybe start with Ed. Ed, can you?
I can hear you. Hi, Susan. Can you hear me?
Yeah.
Great.
Yes.
That was really excellent. I just have a couple of comments and questions. So there's a big, I'm not far from a clinical transfusion person, but I'm certainly aware that there's a big push or a trend in North America and certainly in less developed countries to use whole blood transfusions in trauma, and as well as tranexamic acid in particular, because it's inexpensive and, anecdotally probably, but I think so. Certainly clinical trials that there's a value. So I just wonder, one, are you applying if you're seeing patients, say, after they've received whole blood or after using a tranexamic acid, are you incorporating that information into your studies? And the other question is, I can really see huge value in serially monitoring patients who are currently being treated by, quote, state-of-the-art practices, because you're not going to I think you're going to be challenged by changing practices based on microfluidics. But I can see that you could change practices if you could show serial changes. So, for example, if a patient gets whole blood transfusions, and then maybe there's a time to change to platelets or this or that or the other thing that correlate with your technologies.
Yeah, great questions. And thanks for joining online. And now I did not see you guys, but now I see you. And hi, Erin, thanks for joining. Yeah, like I said, great question. So your first one, when you're doing a bleeding patient study, this window of time that you're talking about, making sure if you're getting a sample before or after transfusion is really challenging. So the way that we do it at our institution is we get samples at arrival, not at hospital admission, but at arrival. So when they show up in the ED, we have very different, different EMS practices even within our own city. So the vast majority of patients that we get samples from at arrival will not yet have received a transfusion. So we're mostly avoiding that confounder. Some of them will, and we know about it, and we can look at that because that data is collected at our institution, but that data is certainly not collected at every institution. And it's often very difficult to get transfusion data. So that's a big challenge, but we factor it into our analyses. You also have to have a very severely injured and bleeding patient to get a transfusion as well. So a lot of these patients that have lower injury severity won't get a transfusion at all. But it's definitely something that is a big challenge to collect. You have to monitor it and factor it into your analyses. And again, as I said, we typically avoid it. We have one study that we've done, and that paper was actually just accepted, where we got samples and we did microfluidics pre and post transfusion in TBI patients. And this is just a small anecdote to kind of get at some of the nuance of your question. And what we saw is pre and post platelet transfusion in TBI patients, you could detect it not at all. Not in microfluidics. You couldn't detect the effects of the platelet transfusion functionally in microfluidics, nor in TEG platelet mapping, nor in aggregometry, nor in flow cytometry. So, I mean, I think there is still a big existential question of like, what the hell are transfusions doing in these patients? And is it functional? Is it volume expansion? Is it related to the endothelium? So I think we're thinking about all of these things and trying to design studies appropriately and thoughtfully. And your points about whole blood and tranexamic acid, I think, are also good. And I didn't talk about those either, but whole blood is becoming more and more common. It's certainly still not everywhere. And TXA is becoming also pretty much ubiquitous. I think most, I mean, there, so, and a correlate to that point, and I'll try to get to the point, but in some trials, you can't even do a trial now if you, some centers won't even do a no TXA trial because they view it as unethical, right? And our group and others have looked at the effects of TXA on that venous thromboembolism signal, but really by and large, by high meta-analyses, TXA is safe and saves lives. So we're doing some studies now where we're going to do some ex vivo simulations with TXA, and I mean, I think the big answer to your question is really just like we need more data, right? You need high end, you need a lot of patients, you need some patients that have gotten transfusions, you need some patients that have gotten TXA, and you need some that don't, and that's actually pretty impossible to do at a single center. And so I think one of the bigger things is we really need to look at collaborating across centers to capture all of these patients to be able to tease out all of these nuances. And I actually, what was your was that, I forget what your second question was.
That was it. No, you sort of covered it. No, but sometimes I think I just wonder about, you have to make a decision when going with the flow and say, okay, fine, everybody's getting TXA. Let's see what it does. And as opposed to trying to come up with a story not to use it. You're not going to do that. The clinical data is too strong. So go with that, use that as your base, and then see what other things change. That was really my point. Anyway, very good.
Thank you. Yeah. And right, you're talking about the kind of following up and showing differences over time, which I think is really interesting. And I also think, just to close, looking at the right endpoint also matters. And I think longer term endpoints, there's some recent trials too, and in COVID we saw some of that, where some of the trials started looking at outcomes 35 days later, or 30, like a month after patients got out of the hospital. And I think especially in the bleeding patient cohort, we have to start focusing on that too, because these decisions that you're making in the first 24 hours, in the first 5 days, actually probably have implications 2 months later. And we're totally not capturing that whatsoever. So lots to be done.
Thank you.
Thank you. Oh yeah, Aaron, or
Aaron, we could take your question, Aaron. Online.
We can hear you, Aaron.
We cannot hear you.
Can you hear me now?
Yes.
Can you hear me?
Yes. Can you hear me? Okay.
Yeah, very interesting talk. Thank you. My question is about the device of this. Is that possible to make multiple of those, or make them in larger scale, so these could be used as for screening purposes in blood supply centers?
Yeah, that really, really great question. And so I think you're spot on. I think that device in particular, the bleeding model that I showed, is a bit high volume. We make about 8 of those devices per single microscope slide, so you get some high throughput. But if you were designing a model to be a screening tool, we would probably make some different design choices and make it smaller volume, higher throughput, faster endpoints, easier to read. But I think that's that's exactly where I think we'd like to go, and we're submitting this data to the FDA so that they can consider whether some flow-based assay, whether it's this assay or another assay, should be a part of the regulatory discussion. I think is prudent. But yeah, to your point, that device in particular is really well suited to bench research and probably not scale up, but there are other design choices that we can make and other models that we can use that would be really well suited to high throughput.
Thank you.
Yeah, thanks.
Thank you. You can hear me, right? All good. Awesome. Thank you for the talk. I just had a question kind of about the changes throughout storage, and sort of what you were showing about the cold storage and in crystalloid media, and kind of what you were showing about the taurine as well. Like, it seems like the metabolic quality of the cells is pretty important for the storage quality and for their subsequent function. I'm wondering if you what do you think is going on? Like, what is it that you think that these cells have going right for them, be it in cold storage or in the crystalloid media or whatever it is, that the room temperature ones are not doing properly?
Yeah, great. Really interesting point. So you're exactly right. In storage, it's a little it's complicated more than just maintaining, kind of keeping a cell alive or another cell that doesn't have clotting function. Because when these cells and when these proteins are functional, they clot, and you actually don't want them to clot in the bag. You only want them to clot in the patient. So you want them to be keeping themselves alive, and that requires metabolism. So you want to give them like just enough so that they can maintain themselves without them being too functional and clotting. And that's a really fine balance, because what we're also seeing is that these products that have probably the highest function at baseline may or may not store better because they're going to clot during storage. So you probably want like maybe a middle of the road product. Maybe it's the middle of the road platelet population that is going to last the longest and still be functional upon transfusion. So it's a really interesting problem. And then when I was talking mostly about platelets, but then if you think about whole blood storage, right? Red cells and platelets like different things. And actually, if you give red cells oxygen, they'll do their job, and their storage lesion will progress faster. And if you store them in an anoxic environment, they're more quiescent. But if you store platelets in an anoxic environment, you've killed their ability to maintain their metabolism. So it's a very complex, I think, set of constraints that's probably different for different aspects of these blood components. And we still, I think, have not optimized storage, right? We're also storing platelets at 4C. Is 4C the best? Is it 2 or 6 or 8 or 12? We don't know. And we're showing them at 4C because that's the temperature of the fridge, right? So, I mean, there's tons of optimization that has never been done that could be done. And so with the taurine study in particular, right, we were just trying to identify which of these pathways were most important for function. And if we can play with them. So if there's one pathway that's really getting consumed, can you block it or can you add something else? And it turns out we can tweak function by looking at that. But it's probably, I mean, there's different things for every part of it. And then your next question was about cold storage. So when platelets hit the cold, they have cold sensitive receptors, as I mentioned. And those receptors actually cause cytoskeletal rearrangements to make them more procoagulant. And so they do, if you measure the cold versus the room temp, and this has been done over the last 10, 15 years, those cold platelets just at baseline, just by being put in the cold, are more procoagulant and have higher activity. So this is why the military is so interested in this and why we're so interested in it for bleeding patients, because by putting them in the cold, you have a more prohemostatic product, so you might have a better product for your bleeding patient. But that comes with those same challenges. They aggregate more in the bag, right? They are activated, they're releasing their internal contents. So it's definitely a balance between all of these different things and also trying to optimize their function at the very end and not necessarily in between. That answered your question.
Sure. It's a wonderful talk. Thank you. The great attraction of your model is its simplicity, but of course that is also the potential weakness. You have no endothelial function and things like that. Yeah. There is quite a lot of information on it. It used to be frog mesentery work, plus some work on rabbits. You can render animals thrombocytopenic using antiplatelet serum. Have you thought about trying to get some form of simplified animal model so you can actually the attraction of your thing is it's I wouldn't say it's easy to do, but it is clean. And if you could tie that in with some of the in vivo models, I think that would be really a terrific advance. But the alternative is to forget in vivo models. They're complicated because I've been involved with them, and just take your data and the clinical data and correlate it and miss out on the middle bit. What are your thoughts?
But yeah, so we're doing both.
Okay.
Yeah, really well received. And I think as what ultimately made me a better scientist in training was working with clinicians and being challenged not just on building a device that has a really nice dose response curve, right, but validating it against an animal model or a patient. So a lot of what we do, and I'm focused on the bench, but I have close collaborators at my institution where we'll go into animal models. So we have murine bleeding models, rabbit bleeding models, mostly small animals. We have another study, that cryoprecipitate study I showed. That product is being tested in non-human primates in a bleeding model. So we are trying to pair this high throughput, as you mentioned, pretty simple, straightforward data that we can get at the bench to provide insight into these mechanisms, whether it be with a paired animal model that actually gets at a bleeding endpoint, right? Or if we get to the patient outcomes as well. So we're going in both directions.
Thank you.
Thanks a lot, Susan. That was really interesting. I was curious about the lower platelet deposition you saw in your microfluidics. I swear that I've read a few places that thrombocytopenia is pretty common. You said that it wasn't, so I stand corrected, but I'm sure that I've read it in a few places. I was curious in your models, is it the plasma or the platelets that's messing things up? And have you started to separate that by putting TIC platelets in with normal plasma and TIC plasma in with normal platelets?
That's aim two. So yes, and so the thrombocytopenic question, bleeding patient, like very severely bleeding patients will have consumption, will get thrombocytopenic, and there's other bleeding etiologies, but the vast majority of trauma patients, whether they be blunt or penetrating, aren't showing up at the ED thrombocytopenic. They get thrombocytopenic, and you probably have some odd patients, like, if your patient is massively exsanguinating, everything is going to be low, but your kind of run-of-the-mill fall, car accident, not massively hemorrhaging patient is actually not thrombocytopenic, despite there probably being lots of consumption happening, right? So it's a bit of a conundrum, but they're dysfunctional, which I think is also is not something that's necessarily captured, or it's captured, but it's not very clinically useful because everybody is dysfunctional. So plasma or platelets being the problem, we don't know, but there's probably soluble mediators. It's probably a little bit of both. So there's a concept in trauma and in bleeding patients where you have basically ghost platelets circulating. So they've activated, they've released their granules, they've released their contents. They're still circulating. They're still going to show up on your hematology analyzer, but they're probably not very functional. Or you have all of these cytokines and DAMPs that are being released, and there's a ton of soluble mediators that are bound to your platelets that are blocking them. Same thing in stored products, I think, right? Could be the soup is what's, you have lots of cellular content that's released over the course of storage that's inhibiting things and maybe going to inhibit your patients when you transfuse them as well. But I don't think we know. So what you've described, we're actively working on that. And there's a couple people have shown that trauma plasma alone causes endotheliopathy in vitro. You can put that on endothelium and, they get all dysregulated and they get leaky. And then there's one or two other groups that have shown if you put healthy platelets in trauma plasma, they lose aggregation responses. But why that's happening and what is the key soluble mediator, we don't know. So we'll definitely be doing those studies, both with resuspending trauma platelets in healthy plasma, vice versa, and in bringing in the blood product and understanding the role of what the contents in the blood product are doing as well.
Is your tissue factor looking the same before and after the experiment?
That's a good question. On the surface of the device, haven't looked. If you have ideas, let me know. We can look at it. Yeah.
Any other questions?
Thank you. Oh, not too much.